So this will be the second part of molecular interactions, or the chemistry portion, our chemistry type lecture. Basically, we started talking about biomolecules. Again, there are four major. We're talking carbohydrates, lipids, um, proteins, and nucleic acids. So basically, we're going to be describing these, kind of how they are created and so forth, and somewhat of their functions in the body. Right. So first off, we're going to start with the carbohydrates. Here's a little bit of what you've learned. These are questions you might want to look at. To kind of see if you're grasping some of the concepts. Again, use a study guide for your studying for the exams, but this is just giving you a little bit of kind of making sure you conceptually you get the stuff from the previous part 1 lecture. It's not all, this isn't all encompassing, but this is to try to intrigue you guys to make sure you are grasping the concepts. If you can answer some of these, probably getting a good grasp of the concepts, and but again, make sure to use the study guide for studying for the exams. And we will move on here to those carbohydrates I was talking about. Okay, carbohydrates. You can see the general formula is you have an N amount of carbohydrates or carbon in carbohydrates, and then 2N of hydrogen and N of O, so CH2O. That's why we carbon with water, carbohydrates. Okay, your basic ones that you guys have seen is glucose, galactose, fructose. Simplest, C6H12O6. Okay, if you look at these, glucose, galactose, fructose, they have the same chemical formula. They are all C6H12O2. What there is just a simple difference in a bond. Look down this column. What do we see? Only one hydrogen bonded on this side, where on galactose there are 2 hydrogen. And then with fructose, you got this double bond here. Okay. And that's going to give you chemical difference. Your liver is good at processing glucose. It has a harder time processing these fructose. So this simple change in one bond makes a big difference physiologically. Okay, you've heard of high fructose sugar. If you guys listen to dietary recommendations, fructose is hard on your liver. It taxes it, where glucose, it has a much easier time to deal with. Okay, so these simple changes in chemical bonds can have drastic effects in the body. Okay, or change the game. So organic carbohydrates, going back, we have galactose and glucose. Okay, those are called monosaccharides. Okay, the individual ones. But typically, much of our food, we don't have glucose simply as glucose. Much of our foods, we have these longer chain of carbohydrates. Carbohydrate bound to carbohydrate bound to carbohydrate. Okay, a monosaccharide, multiple monosaccharides. Okay, a molecule in which 2 monosaccharides, here we got galactose, a monosaccharide, glucose, a monosaccharide, bound together creates a di-2 saccharide. All right, common sucrose is glucose and fructose. Okay, we got lactose, maltose. Maltose is 2 glucose. This would be lactose here, glucose with galactose, and sucrose would be glucose with fructose. These are your common disaccharides. Okay, and we'll see how this bonding, how these chains are formed, what are the chemical reactions that take place that form these chains. Okay. Organic molecules, now we got multiple, 3 or more, and we get a polysaccharide. Okay, we start binding these together and we get these long chains. If we put another monosaccharide here, we'll start building these chains. Your glycogen in your blood or starch in potatoes and bread, those are long chains of carbohydrates. Okay, they are polysaccharides. Okay. So let's first look at how these formation of these chains of carbohydrates take place. All right, look, we got glucose and glucose. Okay, 2 monosaccharides, and the equation goes to form maltose. And you can see where this bond is forming, here to here, and what gets created when this bond is made. Okay, the molecule basically gets dehydrated, water is formed. Water is taken out from here to form this bond. Okay, so therefore these equations, or sorry, this chemical reaction is called dehydration reaction or condensation reaction. We will see with many of our biological molecules, our proteins, our lipids, they are formed via these dehydration or condensation reaction. When they bond together, water is formed. Okay? If we were to add maltose and bind maltose to maltose, we would get a 4 chain, and now we would be at a polysaccharide. But again, each time we bond a monosaccharide together, a molecule of water is formed. Therefore, these reactions, dehydration or condensation reactions. Okay, let's look at the opposite. The breakdown of carbohydrates. Many of our carbohydrates we eat are long chains. We eat potato, bread. They are chains of carbohydrates. They're not just monosaccharides. They are these long polysaccharides. So what we do to form the bonds between these monosaccharides, or to form these chains, is water was created. Now to break it down, we need to add water to these bonds to basically cleave them, to cleave these bonds, to lyse the bonds. We need to add water. So these reactions are called hydrolysis. We're adding water and we're lysing the bonds between, in this case, we have starch, which first break it down into the disaccharide, maltose. Okay. And then ultimately in our digestive tract, we're going to break it down, maltose and water, and break it down to the individual monosaccharides. Okay. So to form condensation or dehydration reaction, those are synonyms for each other. And then to break down carbohydrates, hydrolysis. And you're going to see these reactions are what take place in these other biological molecules to form them or to break them down. So next up is, in biomolecules, is our lipids, our triglycerides. And insoluble in polar solvents, right? That's that analogy when we drop oil, which is a bunch of lipids, into water. What happens is they stick together. They don't dissolve in the water because lipids are these long chain of carbohydrates. And this long chains, even though there's oxygen here, there's these long chains of carbohydrates. It allows for the electrons to be evenly spaced, and allows for these molecules to be nonpolar. Okay, so these fatty acids that are going to help make our triglycerides, these long chains of carbons, have a nonpolar characteristic to them. Therefore, they don't like to mix with that polar solvent of water. Okay, but how do we make a triglyceride? Tri meaning 3. We have fatty acid. Fatty acids are these long chains of carbon with a carboxyl group, right? Carboxyl functional group making it an acid. Okay, long chains. So we get 3 of these fatty acids, and they bind to a glycerol. Okay, a 3-carbon backbone here. You can see fatty acid bind here, fatty acid binds here, and what do you see happening? What gets formed as this bond between the fatty acid and the glycerol takes place? We form water. Again, we're doing a dehydration or a condensation reaction to form these triglycerides. This is how your fats get stored. They get stored as these triglycerides. Okay, and then if we need energy, we can break these down, form our fatty acids again, hydrolyze it, and then we can use these, we'll see later, we can use these fatty acids to create energy. We can use them. So it is and we'll see we can get a lot of energy, a lot of ATP from each of these fatty acid chains. So it is a good storage molecule for when we need energy. You may have heard the terms saturated and unsaturated fat. Since we're doing physiology, we're talking about homeostasis. Diet comes part of that. And you can see the difference in the chains, the saturated versus unsaturated fats. Here is saturated. Okay, palmitic acid. Do you see any, along this chain of carbons, do you see any double bonds? No, they're all single bonds between the carbons. That means it's saturated. In an unsaturated fat, what do you see? You see double bonds between these carbons. The difference is, chemically wise, like olive oil is essentially all fat, but it is made up of unsaturated fats, where lard is made up of saturated fats. Remember our van der Waals forces. We saw those saturated fats. This is a very linear molecule. Is it easier to stack linear next to linear? Yeah, they can stack together, and therefore, what do you see when you see lard at room temperature? Okay, it's that sticky substance, but it's not an oil, it's not a liquid. Where do you see olive oil? That is made up of unsaturated. So you got to think, it's not shown here but there are these, so instead of being very linear, there's these kinks in the unsaturated fat. These double bonds, 3-dimensionally, make kinks in it. Those are harder. You can't stack them one on one. And therefore, at room temperature, we see unsaturated fats typically as a more liquid form. So chemically, that's what's making that take place. Another term, trans versus cis. In real life, in the real world, typically most of our natural fats are cis fats. Okay, cis, you can see the difference between, okay, oleic acid, which is cis. See that double bond there? Okay, carbons there. Lactic acid, which is a trans fat. Okay, the hydrogen are on opposite side. And this just basic chain in bonding, this basic change in the structure, even though these, count them, they have the same amount. This basic change in structure has a drastic effect health-wise. You've always heard, don't eat trans fat. Trans fats, typically fats are heated up to create these trans fats. It's great because they stay well for a long time, problem is your liver can break this down. It has a easier time. It has the trans fat taxes your liver because it has a harder way to deal with these trans fats. Okay, so there are even just these small little changes in chemical bonding have a drastic effect come, say, health-wise. Okay. Ketone bodies. You remember ketones? We have a carbonyl. That in the middle chain we have C double bonded with oxygen. Okay, this one is also a keto acid because it also has a carboxyl group on the end, acetoacetic acid. Okay, it is a ketone along with an acid, an organic acid. Okay. Where do these come from? The ketone bodies, where do we see them? Those of you that heard of the ketogenic diet, basically what you are doing is increasing the liver to form more ketones. Okay, ketones can be used as a source of energy. Many of your cells can use ketones as an energy source to make that ATP we'll be talking about a little bit later in the semester. Okay. When you have lots of fatty acids, so when you do a ketogenic diet, all you're basically doing is you're eating more fats. So your body doesn't have glucose. It has glucose, but you're not feeding your body constantly with glucose, and you're also lowering your insulin levels that allows fat to be broken down. You can access those stores of fat. When you break down the fat, put it in the blood, when you have lots of fatty acids in the blood, your liver will convert them to ketone bodies. And that is why as you break down more fat, you find more ketones in the blood. In a ketogenic diet, that's what happens. Your liver is converting those lipids into ketones. Okay, you can see this is an acid. What do you think happens to your pH in a ketogenic diet? Think it's on the more acidic side or the more basic side? Yeah, it might drop a little bit and become a little more acidic because now you're putting in these ketones, these acids that weren't there before. It doesn't get pathological. Okay, we will see in diabetes, if you don't treat diabetes, diabetics would have high ketone levels, and we'll take a look at that a little bit later in the semester. Okay, the ketones are basically fatty lipids, fatty acids. Your liver, if there's high amounts, will start converting them to ketones, and ketones can be used as an energy source to create energy. And one specific fatty acid we want to take note of is one called phospholipids. Okay, you can see a lipid along with phosphate, so you can see there is a phosphate group here. Okay, here's your glycerol background. There are two basic chains of fatty acids, right? And then your phosphoside. This gives this molecule amphiphatic means there is both a polar in what we call the polar head to the molecule, and then there is a nonpolar side. The fatty acid chains, nonpolar. Okay, so it has both a nonpolar characteristic and a polar characteristic. You can imagine, which side do you think faces water when these are put together? Yeah, which one's like the water? Yeah, the polar side. These side, the fatty acid chains, nonpolar, don't want to combine with water. So we're going to see these make up a big component of, or make up basically a component of the cell membranes. Okay, that barrier around the cell. We're going to see it's going to make a lipid bilayer. Basically, these polar sides will be attracted to each other, and then the outer that leaves the outer polar sides in contact with those solutions, that watery environment. Okay, so when we look at the cells, when we start doing membrane transport, we're going to be looking at phospholipids, some of their characteristics. But again, remember there is a polar and a non-polar side to these phospholipids. Another molecule to take note of that's part of the lipid family are steroids. You see they don't look like the classic triglycerides with those long chain of carbon atoms, but they are very nonpolar and insoluble in water, and those are the steroids. Okay, classic is one you know or have heard of quite readily is cholesterol. Okay, you can see the structure here with these 4 rings structure, and cholesterol is, well, you always hear it in the bad sense, the bad cholesterol clogging up arteries. They are also, it is a precursor to many of our hormones, our non-polar hormones, like cortisol. See here, just a cleave of this, adding some bonds, and you end up with cholesterol. Probably all heard of testosterone, estrogen. They are the precursor to these is just cholesterol. We get modification of that cholesterol molecule through, we'll see multiple steps, but that is the precursor to these steroid hormones. All right, one of the major players in biology are our proteins. Okay. Our proteins basically are built by those amino acids. And when we already saw the functional group for amino acids. What we see for all amino acids is all amino acids have a similar backbone. They got the amino group, carbon, and then a carboxyl group on the other end. What makes each amino acid different is the functional group that's bound here. Okay, you can see we have non-polar, polar amino acids, and the polarity comes from what functional group is bound. Okay, look at the non-polar amino acids. Functional groups, carbon chains or aromatic ring here, these are very non-polar characteristics, giving the amino acid a non-polar characteristic. But what do you see similar? They both have the same chain here. They both have the amine group, the carbon, and then a carboxyl group. The only difference is what's taking place here in the functional group. Polar amino acids, you can see they have that same amino acid backbone. They just have a different functional group up here. In this case, it's polar. We can have basic, okay, or we can have also acidic. So that R group gives it its functionality. And what we see is basically our proteins, just like our carbohydrates. We had monosaccharides bound to monosaccharides, and these long chain of monosaccharides bound together gave us our polysaccharide. In this case, our proteins are just basically amino acids bound together. The amine group of one is going to bind to the carboxyl group of another. So this would be, say, this amine group could come up, form a bond here, and we get these long chains of amino acids, which basically makes our proteins. But what you can see compared to those carbohydrates is there are 20 different amino acids. So there are 20 different functional groups with very different characteristics, polarity wise, basic, acidic, and so we can get these interactions between these functional groups, and it also gives these proteins a very diverse complex to make these diverse molecules, and basically the proteins are the functioning molecules in our body. You think everybody's heard of DNA. What essentially is DNA? DNA are the codes to make the proteins. All our cells have DNA because we got to make these proteins. These proteins are basically kind of the main players. They're the ones that are going to be doing the work, the functions through the body to make sure everything runs smoothly. And we can change how much of these are around by changing how active we're decoding our DNA. Okay. What do you think kind of reaction when we bind this amine group to this carboxyl group? What kind of reaction do you think is taking place? Yeah, dehydration condensation reaction. We're going to see we form what's called a peptide bond between those two amino acids. Okay. Also, one thing to note, you'll hear essential amino acids and non-essential amino acids. We have 20 different amino acids to build from. There are essential and non-essential. Essential means we have to eat them. We have to get them out of the diet. We cannot make them. We don't have the enzymes to make the amino acids. The non-essentials, we could take other molecules and essentially form these non-essential amino acids. So our bodies have the ability to make these, so we don't technically have to get them through the diet, but we must eat our essential amino acids. We have to get them through the diet somehow because we cannot make them. We don't have enzymes that can take another molecule and turn it into this amino acids, but we do have enzymes that can convert certain molecules and convert them into these non-essential amino acids. So that's the difference between essential and non-essential in that regards. Okay, so primary structure. So what we see when we say our proteins. Our proteins are basically long chains of amino acids. You can see a carboxyl group of one amino acid is bound to the amine group, or bound to the, and we form water. So we do this condensation reactions to form, this is a special name for this bond called a peptide bond. Therefore, many times you'll hear proteins called polypeptides. This is a long chain of amino acids. Okay. This chain, what amino acids? Okay. Depending on what protein it is, depends on what amino acids are here. Okay, that's what your DNA tells you. What is the order in which the amino acids are bound together? That essentially is all the DNA is saying, okay, this tyrosine needs to go here, which is an amino acid. Alanine needs to go next. All right, threonine needs to go third. That tells you what amino acids need to go in what order. This gives us the primary structure of our proteins. What is the order of amino acids bound together? Okay, because what we're going to see is these amino acids, or sorry, these proteins are these three-dimensional structures. The first level of structure, primary structure, is just what is a chain of amino acids? Because what we're going to see is there's going to be interaction between these amino acids in these chains, which will give this protein folding and give it its three-dimensional structure. All right, secondary structure is, we will see, okay, alpha helix or beta sheet. Hydrogen bonding between the carbonyl and the amine hydrogen give us either this helix structure, okay, or you can see the hydrogen bonding here gives us this pleated sheet. Okay, so this is the next, now we're starting, we had this chain of amino acids. Now we're starting to get this three-dimensional structure here. The secondary structure is alpha helix or beta sheet. Some have all alpha helixes, some have all beta sheet, some proteins have both an alpha helix in one area of the protein and a pleated beta sheet in another region of the protein. Just depends on the protein. But now we're starting to give this protein a three-dimensional shape. Now we start getting what's called our tertiary structure. Tertiary structures typically formed by interactions between the R groups of those amino acids. Okay, you can see here, you get ionic, and these interactions can be ionic bonds, hydrogen bonding, van der Waals forces, okay, and also covalent bonding. So we start getting these interactions. So the key is we got to make sure we have the right amino acid in the right location. Here you can see we got a nonpolar with nonpolar to give us a van der Waals force. If for some reason the amino acid got changed here and we didn't have this R functional group, we may not get this interaction. And what might happen to our protein? Our proteins are key that we have this proper three-dimensional structure. If we didn't get this, if this amino acid was changed here, that could change the interaction here, and we may not get that fold in the protein that we need. If we don't get that fold, there's a good possibility that protein will not have the proper function. Okay, that's where we get mutations. Remember, the DNA tells us which amino acids need to go where. In a mutation, that's a change in the DNA. If the DNA is changed and this amino acid gets changed in the code, you can see we may not have a functional protein. Okay, because we got to have this interaction to give us the proper tertiary structure, the proper three-dimensional structure for the protein to be functional. Okay, so all proteins, we get up to tertiary structure. Some proteins even have quaternary structure. Okay, most have tertiary and they have this three-dimensional each protein, but some proteins, you can see here, each of these colored are a protein, and there are 4 proteins that come together to form, this is our friend hemoglobin. Okay, and so you get this quaternary structure. Okay, besides just the three-dimensional tertiary, when they move together, they form a quaternary structure to give the full functioning protein. Not all proteins have quaternary, some are just individual proteins that have their tertiary structure, but again, just stressing that it is the three-dimensional structure that is important. If this three-dimensional structure gets moved in any way, or gets what we'll see process called denaturing, where it unravels and you don't get this three-dimensional structure, then the protein's non-functional. Okay. One way protein's denaturing, you can see here's the normal protein. You can see how we got the primary structure is just the strand of amino acids. You can see some alpha helixes in here, the secondary structure, and then there's further folding to give you that tertiary structure, to give you this three-dimensional shape. Loss of when we denature, one of the things that can denature a protein is high heat. Okay, extreme heat can cause disruption in those interactions. Right, that tertiary structure, we have interactions, and also secondary structure, we have interactions. You heat it up and it may disrupt those interactions, and the protein might unfold, or what we call denature. Okay, also pH. If it's too high or too low, that can cause disruption in interactions between those either hydrogen bonds or those other bondings, those other interactions, and we can get, again, denaturing of the protein. So you can see why we need to keep our temperature regulated, we need to keep our pH regulated, because these proteins, and we're going to look at all the different types of proteins through the body, what their functions are, but these proteins are basically the functioning molecules. If they get denatured, your cells can't function because they won't have the needed proteins if we have these needed proteins to do their functions. Okay. One note, doctors recommend pregnant women don't spend long stretches in hot tubs. Okay, many proteins are enzymes that help catalyze those reactions. You think in a child or a fetus that's being developed, there are all these chemical reactions taking place to make sure development takes place properly. So you don't, I mean, this would be extreme case if the woman sat in there for hours and hours, her core temperature could get high enough where it could probably cause some disruption in certain proteins in the developing fetus. Okay, so this is important. Can't have high heat, can't change pH. These are factors that all affect the functioning of proteins. And just a rundown of the structure. This gives you primary structure. Okay, just a strand of amino acids. Okay, how are the amino acids attached? In what order are the amino acids attached? Then we get secondary. We can have alpha helixes or beta pleated sheets. Okay, some proteins have both. And you can see then the tertiary. Then we get further folding, those interactions between those R groups, those functional groups of the amino acids, and we get that further folding into the tertiary structure. Okay, again, some proteins only have tertiary structure. Some proteins come together and they will form a quaternary structure. Okay. But that gives it its functionality. It has to have this proper folding to have its functionality. Many proteins don't exist just as proteins. Many proteins aren't just amino acid strands and folded up. They also will have some conjugation, such as glycoproteins. See, here's the blue is the protein. Okay, this is on the cell membrane, and we have carbohydrates that are tethered to, basically we're like tethered to them. Many of your molecules that designate what a cell is, or surface markers, are glycated proteins. Okay, one you probably have heard of, lipoproteins. Lipid proteins, right, are those very low density lipoproteins, those cholesterol. Okay, good and bad cholesterol you probably have heard. Here they are lipoproteins. They are proteins bound with lipids. Okay, brought together. See the bad, the LDLs, and the good, the HDLs, high density. Okay, but again, these are proteins, but in this case, they are bound with other molecules. Glycoproteins with carbohydrates, lipoproteins with our lipids. Okay, we'll talk a little bit more when we get to heart, so forth, on these lipoproteins. So, like I said, the proteins are the functioning molecules in the body. They are what makes things go. They are the ones that get the work done and do the functions to make sure the cells can operate properly. Okay, you can tell how important they are because our DNA is basically codes for all these different proteins, and we have roughly up to 20,000 genes that code for all the different proteins. And we'll see there's even more besides just 20 proteins. There are 20,000 proteins. There are multiple proteins that can be coded for each gene, giving us a large amount of proteins that can be made in our body through our codes in our DNA. Okay, but what are some of the functions? One we're going to touch on enzymes. Okay, we'll see enzymes in a bit. We already touched on enzymes are those molecules that catalyze reactions. We're going to talk about these in the second. For the second exam, we'll be touching on enzymes because we'll be starting to talk about cellular respirations where these come into play quite a bit. First exam, membrane transporters. Okay, these are transporters that are in the cell membrane, embedded in there, allowing molecules to go in or out of the cell. Okay, we will spend a good amount of time talking about these because we will see them throughout the semester. Signaling molecules, you can think signaling molecules, where will we be talking about these? All right, the nervous system and neurotransmitters. Some neurotransmitters are proteins or peptides. Also hormones. What do signal molecules bind to? To signal, they bind to receptors. So again, nervous system, endocrine system will be the big ones when we talk receptors. Okay, binding proteins, usually on the surface of the cell, help the cells move around and stay tethered or bound. Immunoglobulins, the one that we've been talking about in the news, those would be antibodies, would be immunoglobulins. They are proteins that are made. And then also regulatory proteins. Okay, so you can see these proteins are important because they allow the cell to do lots of different things, allow them to serve their function. We got to make sure, keep them properly folded. So hopefully I've nailed into you the importance of the proteins having this three-dimensional structures. Okay, because many of these proteins, since they are the functioning parts of the body are functioning molecules, they have interactions with many other molecules. So to have this proper three-dimensional structure, because many of these proteins interact and they interact at the areas where they have a binding site. So this area has to have the right amino acids in this binding site, the right configuration, the shape, to make sure that these interactions will take place. Okay, they form these noncovalent molecular interactions between what we call a ligand. Okay, ions or other molecules that bind are called the ligands. In an enzyme's case, if the enzyme was an enzyme or a membrane transport, these molecules would be considered what we call substrates. So kind of remember that terminology because we'll be using it throughout the semester. Okay, and these sites are usually specific to a single molecule or a kind of class of molecules. Okay, they don't just bind everything, all the molecules out there. They are specific for a certain molecule or a certain class of molecules. And when they bind, there's this induced fit. Basically, as the molecule moves in, the protein kind of moves in to fit to help create these molecular interactions, okay, called the induced fit model. Right? There is another term that will be seen a lot through the semester, and that is called affinity. Okay, it describes the degree of attraction. How tight is that fit? Okay. Greater affinity, all right, means the protein binds tightly, doesn't want to let it go as easily. With a lesser affinity, it's a weaker attraction and it's more willing to let the molecule go. We'll see hemoglobin is one of those molecules that has differing affinities depending on where it's at and what is in the solution around it. Because we need hemoglobin binds our oxygen and carries it through our blood. At the lungs, we want it to have great affinity. We want it to bind that oxygen so it can carry it through. But when we get to the tissues, we want oxygen to release the oxygen. Okay, so we want it to have lesser affinity, and we'll see how that takes place. But just remember for now, affinity, great affinity means tight bind. Lesser affinity means a looser bind and more willing to let whatever ligand or substrate is bound to the protein. Okay. So the last, we're going to touch on the nucleic acids because we're going to move into protein synthesis. Luckily, protein synthesis is going to be a little bit shorter lecture than this chemistry one. Again, I try to keep the chemistry one, try to be as specific as possible because I know some of this, I know this stuff's important because we're going to be using it, and some of you, this is where kind of not the wheels fall off, but start to struggle a little bit. So I want to make sure we get it nailed down so we're clear throughout the rest of the semester. Okay, but nucleic acids, this is the last of the 4 biomolecules we'll be talking about. And as we've already stated, it houses that genetic information. That genetic information is simply the codes for the proteins. Which amino acid and in which order are the amino acids put to make all these thousands and thousands of proteins that we can make in our bodies. Okay. The nucleotides, that is these here. You can see it's got 3 main components. It's got this sugar, a pentose, and DNA, we have deoxyribose sugar, therefore the D, okay, and a phosphate group, and a base, okay. All those nucleotides of DNA have this deoxyribose as phosphate. What differs is what base is here. And we can see there are for DNA, there are 4 different bases. There's the single ring pyrimidines, that's cytosine, you'll see written as a C, thymine written as a T, and then in RNA, the difference between DNA and RNA is this is a ribose sugar, okay? And RNA, instead of T, it has uracil in the place of T. All right, and then the purines is a double ring nitrogen structure, and those are guanine and adenine. Those are the same in both DNA and RNA. Okay, but these nucleotides are basically the codes. The order in which they are in the DNA helps the code in which amino acid comes and what place the amino acid comes. Okay. So we are going to discuss this in more detail when we go over the protein synthesis, because we got a code for what we call the RNA, the messenger RNA. But this is what's going to, these nucleotides are what hold the code. Okay. So deoxyribose, what did we say? Reactions. So basically we have those 2 long strands that make up DNA. DNA is double-stranded. Okay, RNA is just single-stranded, but the strands are put together and they're made with this phosphate ribose backbone. Okay, if you think of the ladder, sides of the ladder, that is this formation between this phosphate and these ribose. You can see the phosphate comes together, OH, in a what kind of reaction? Dehydration or condensation reaction, because water is being produced. And so this is what makes those strand portions. What makes the rung of the ladder for the DNA is which base is sticking out. Okay, which of the bases are sticking out? Those make up the rungs. Remember, we have 2 strands. So if we had another strand, we would have another base coming out from this strand, and that base would hydrogen bond with this. This base would hydrogen bond with this, and we're going to see this is specific. The complementary ones are specific but that forms the rung in the ladder. Okay? So here is a complementary base pairing between the bases of the different strands. For DNA, we have two strands. We have a base coming out from this one, and we have a base coming out from this one. What we're going to see is there are bases that are complementary with each other. If this one strand has G on the other strand that is making up the run, it will be C, cytosine. So these are complementary. All right, you wouldn't have A over here base pairing with guanine. Okay, you wouldn't have adenine over here. In DNA, thymine, okay, is base pairs with adenine. So if you have thymine on this one, it's going to be adenine on the other part of the rung on this strand. Okay, so if you know one strand, you can figure out what the sequence of the bases is in this other strand, because if it's G here, you know it's C over here. If it's C here, you know it's G over on this other strand. Okay, so just keep that in mind. We're going to see in RNA, it's a little bit different. We'll have uracil instead of thymine. Okay, but we will take a look. In the double-stranded DNA, you can see A is with T, G with C, and vice versa. So these DNA, they are very, very long molecules, these two strands. What we see is they are wrapped up and form these double helixes. Okay, this is like spiral staircase almost. Okay. And although there are only 4 bases, we can make an infinite amount of combinations, and therefore we can have not almost infinite amount of proteins that we make. Okay? Many of the strands contain thousands of nucleotides. So here's showing the differences in nucleic acids. All right. Deoxyribonucleic acid, DNA, has deoxyribose. RNase instead has ribose as its sugar. The other main thing is thymine is substituted with uracil in RNA. And the other main thing is RNA is single-stranded, where DNA is double-stranded. So RNA and nucleotides. So there are several types of RNA. There's more than this, but these are some of the main players in RNA, and the ones we'll be looking at when we start looking at protein synthesis. It gets much more complicated than this, but for our sake, we're going to be looking at messenger RNA, because basically the DNA contains the code, and how we copy the code is we copy it onto what we call messenger RNA. All right, we'll see the DNA are these long strands. There's only certain portions. There are many genes held on this DNA. This one double-stranded molecule of DNA, there are certain portions that have a gene that codes for a protein. And so to make our proteins, we just have to copy that portion of the DNA, and we copy it into messenger RNA. Okay. Another transfer RNA, we'll see these. These are the ones that are going to, when we go to make our proteins, they are the ones that are going to bring in the proper amino acid in the right sequence. It's going to bring it into, say we have tyrosine and alanine. Okay, there'll be a tRNA that brings tyrosine in first, and then a tRNA that will bring alanine in. Okay, so it transfers the the amino acids into what we call what we see here, the ribosome that contains ribosomal RNA. okay, along with proteins. Okay, we're going to get into this more. We're going to look at the actual protein synthesis. Since these molecules are so important, we're going to look at the process of creating these proteins and a little bit on how it's regulated, because they are the functioning molecules of the body. Okay. And lastly, the nucleotides, besides serving as the templates for the amino acids, or the code for the amino acids, nucleotides are found serving other functions through the body. One common one you'll see is adenine, ribose, and what do you see? 3-phosphate. This is our ATP. We're going to spend a whole lecture talking about how we make our ATP, since that is basically the gasoline. Gasoline to your car, ATP is to our cells in our body. It is the it's where we get the energy that we need to do work with. Some cellular regulators, we'll see cyclic AMP used as a second messenger when we start getting into cell signaling. When we go into cellular respiration, there are coenzymes, adenine dinucleotide, NAD. You'll love this molecule when we get to cellular respiration. All right, and then even some are used as neurotransmitters, used as communicators or signaling molecules. Okay, caffeine affects the adenosine receptor, okay, but it affects the signaling done by adenosine. Okay, so they serve other functions. We always think of these nucleotides in case of the DNA, but there are other functions in which these nucleotides operate in the body.